Hello everybody, my name is Sarah Kintzel. Today I will be presenting my project in collaboration with my graduate mentor Taylor in Dr. Scott Gerber's lab. The focus of my presentation will be on defining hypoxia in rectal cancer, a potential mechanism behind radiotherapy efficacy. One of the tumor types that the Gerber lab focuses on is colorectal cancer or CRC. CRC is a devastating malignancy and ranks as the second leading cause of cancer-related deaths in the U.S. Of the approximated 100,000 new cases of CRC each year in the U.S., there are over 42,000 Americans diagnosed each year from rectal cancer disease, which makes up one-third of CRCs. For rectal cancer, one of the current treatments is complete mesorectal excision surgery. However, recurrence is a common problem where about 15 to 45% of patients' tumors will regrow. This greatly reduces organ function and therefore negatively affects the quality of life for these patients. Additionally, while the majority of cases occur in people over the age of 50, there is a recent increase in diagnoses in people in their mid to late 20s, in which the lives are negatively impacted for the remainder of it. Current methods of treatment is radiation therapy, or RT, in which various studies have shown that administering preoperative RT reduces local recurrence and also increases overall survival. RT is a common cancer modality where approximately 50% of all cancer patients receive some form of RT. This therapy targets the tumor locally, killing highly proliferative tumor cells. When effective, it can shrink the tumor size for operation or reduce the need for an excision surgery. One of the key points of the presentation and research done in the Gerber lab is that RT has been shown to elicit a robust anti-tumor immune response. In fact, the efficacy of RT and the response generated is largely mediated by the immune response. Advantages of using RT include a robust immune response, in which vascularization is normalized, which increases immune infiltrate and increases the number of antigen-presenting anti-tumor immune cells, such as the upregulation of cytotoxic CD8 T cells, MHCs, and DAMs. However, the downside to RT is that it is not 100% curative. It acts as a double-edged sword, where it both promotes immune stimulation at the expense of subsequent immunosuppression. I will explain why in future slides. Mouse models are used to recapitulate human disease to study combinations of RT with possible immunotherapies in vivo. We developed a colorectal mouse model where we inject luciferase expressing colon 38 cells intrarectally, as shown here. Since these tumors express luciferase, we can measure tumor growth in vivo over time as a readout of bioluminescence. As you can see, tumors left untreated will grow substantially. Looking at the survival curve, you can see that the mice rapidly succumb to tumor burden around day 40. At this point, these mice have both the primary tumor as well as clinically relevant metastases on the mesentery and omentum. When we incorporate a clinically relevant dosing scheme of RT, where 9 days after the tumor has established, the mice are treated with 5 consecutive fractions of 5 gray RT per fraction. Here I'm showing that prior to RT, the mice are rearranged so that both treatment groups have equal geometric means. Our laboratory has shown that when introducing RT, and we see an interesting response that despite receiving equal treatment, there's a clear divide in RT response. By measuring tumor growth by bioluminescence, we found that whereas the untreated tumors in blue grew as expected, about half the tumors treated with RT had no response and grew similarly to the untreated. However, the other half of the tumors had a strong RT response that led to a long-term cure. We classify this response as the responder-non-responder phenotype. One potential explanation could be related to hypoxia. Previous data established in the lab has shown that colon 38 tumors are largely hypoxic. Here, tumor sections were stained for immunofluorescent microscopy using anti-CD31 antibodies in green to mark vasculature and antibodies that bind to hypoxic regions in red. Reduced oxygen levels in the TME or the tumor microenvironment reduces the ability for RT to create reactant oxygen species to cause DNA damage. This, therefore, reduces the efficacy of RT significantly. The lack of oxygen ultimately dampens the immune response, in which hypoxia is the contributing factor. Immune cells need oxygen to function, unlike cancer cells. Additionally, less blood vessels in the tumor as a result of hypoxia means less immune cell infiltration. Hypoxia influences an unfavorable patient outcome. HIF1 alpha is the main regulator of hypoxia response genes in the absence of oxygen. These survival curves show both disease-free survival and overall survival. In both cases, high expression of HIF1 alpha correlates to worsened patient survival. Additionally, approximately 50% of rectal cancer patients have hypoxic tumors, and the survival rate for patients with hypoxic tumors is around 3 years. The purpose of my study is broken down into 2 sections, the in vitro and in vivo study of hypoxia. My hypotheses include, when colon-38 cells are made hypoxic in vitro, the cells will undergo changes in gene expression and regulation, and rectal tumors will become increasingly hypoxic as their size increases in vivo. Here is the in vitro schematic of the hypoxic experiment to monitor hypoxic gene expression. Here we have 2 petri dishes, one for normoxic conditions at 21% oxygen and one for hypoxic conditions at 0.5% oxygen, which are incubated over 6 hours. The RNA is then collected, cDNA synthesized, and then applied to a standard qPCR assay, in which the data is then analyzed in programs such as CFX Maestro and PRISM. Here are the results from a gene study done in CFX Maestro. The genes chosen here, upregulated in red, downregulated are green, are based on a 3-fold change when comparing hypoxic genes to the normoxic conditions. Here is a bar graph representing those selected target genes based on that 3-fold criteria. Here we'd like to highlight the 3 most upregulated genes in hypoxic conditions. ADM, or adrenomugulin, controls vasodilation and angiogenesis. PGF, or placental growth factor, is a vascular endothelial growth factor which is active during tumor growth. BNIP3, or B-cell lymphoma 2 interacting protein 3, is a gene that encodes a mitochondrial protein that mediates pro-apoptotic and anti-apoptotic factors in proteins. Due to the COVID-19 pandemic, most of this work is largely unfinished. Once it is safe to do so, here is what we would like to do. Determine the expression of and quantify hypoxic regions using EFI, flow cytometry, and immunohistochemistry techniques in rectal tumors of the mouse model. Determine expression levels between normoxic and hypoxic in vitro colon-38 cells of hypoxia response genes at 12 and 24 hours and 0.5% oxygen. Ultimately, by quantifying hypoxia in our mouse model, we can test therapies targeting hypoxic regions of the tumor in hopes to enhance treatment for rectal cancer patients. Here is the proposed protocol for the in vivo hypoxic experiment using EF5 injections. Now, EF5 is this molecule here. It covalently binds to cells under hypoxia and is used and injected into a mouse model. This technique was optimized by the Lord Gerber lab and is commonly visualized using an anti-EF5 ELK-SIDE3 fluorescent antibody in both immunofluorescent microscopy and flow cytometry. Here, this protocol will explore hypoxia in the murine orthotopic rectal cancer tumors. We will inject the colon-38 luciferase tumors into our mouse model and monitor the rectal tumor growth using in vivo imaging software, or IVIS, at days 8, 14, and 24. We will then inject the EF5 molecules intrarectally and harvest the tumors. Some tumors will be then quantified and identified for hypoxic immunosubtypes using flow cytometry, and others will be identified and quantified using immunohistochemistry techniques. Additionally, we would like to determine the expression of hypoxic regions using EF5 in adjacent tissue in the colorectal regions of the mouse model. I would like to thank my principal investigator, Scott, my graduate mentor Taylor, and the laboratory technician Jesse for having this opportunity to contribute to this project and for all of their guidance. I'm excited to get back into the lab when it is safe to do so and move forward with the rest of this project. Please ask any questions in the comments below using the online Padlet format. I know this was a lot of information, but thank you for watching.